my dear students. I hope all of you are doing very well. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. Today, I am going to tell you how we can use microbial biotransformation for our pharmaceutical biotechnology. I hope all of you have enjoyed my previous lecture, where I have mentioned you, I have told you what is microbial biotransformation, what are the various advantages, disadvantages, and how this microbial biotransformation actually helping us to get our desired product. Today, I am going to tell you the applied part of microbial biotransformation. If you have missed this lecture, do not worry, I am going to start you with some basic introduction to microbial biotransformation. Then I am going to tell you how we can use. Basically, there are lots of applications, but I have selected few of the most commonly used application in our certain this area. I am going to tell you about how you can transform steroids or sterol. Then I am going to tell you the transformation of non-steroidal compound, then biotransformation of antibiotics, pesticide, pollutants, and petroleum biotransformations. After that, I am going to tell you something more interesting thing, why our this field is actually growing. Why this field is actually growing, and why nowadays scientists, industry, academician, all the people are actually highly interested in microbial biotransformation. So, what is microbial biotransformation? I hope in my previous lecture you have that is listen and you have so much clear, but in case you have missed, let me that is summarize whatever I have told you about microbial biotransformation in my previous lecture. So, it is a process by which organic compounds are transformed from one form to another to reduce the persistence and toxicity of the chemical compound. Okay? And this process is actually aided by major range of microorganisms and their products such as bacteria, fungi, enzymes. We are actually using these things in microbial biotransformation. Microbial biotransformation also can be used in synthesizing products, that is compounds or material, if synthetic approach is actually challenging. If we do some synthetic activity, if we are going to produce something synthetically and this procedure is much more complicated, or if it is taking much more time or so much expensive, in this case we can use our microbes as microbial biotransformation. You also have seen in my previous lecture that there are two types, either enzymatics or non-enzymatic. Enzymatic are actually also further divided into two way, microsomal or non-microsomal. In microsomal transformation actually caused by the enzyme present in the lipophilic membrane or smooth endoplasmic reticulum. So, what about the enzymes present in the lipophilic membrane or smooth endoplasmic reticulum, ER reticulum, those enzymes are actually using for enzymatic biotransformation. Or non-microsomal, basically enzymes which are present in the mitochondria. If we use mitochondrial enzyme, we will call it as a non-microsomal. Basically enzymes are involved in both these cases. Another thing I hope you can remember, I have told you about the non-enzymatic. Here enzymes are actually not used, it is actually that is taking place at a physiological page. Some of this includes like these are the few example where I have mentioned that how we can convert all those substances into their new product, like this thing called chlorazeptate or mustane or atracurcumin, and then this thing called ludanosine and hexamine. We can convert this thing into other product, like we can getting here for this thing called dimethyl diazepam. Let me remove the ink. From chlorazeptate we got the dimethyl diazepam. From mustine HCl and that is converted into the this thing called ethyl nimonium. Then atracurcumin is converted into this thing called ludanosine and quaternary acid, hexamine converted into formaldehyde. So, we can get these things by using microbial biotransformation. Basically non-enzymatic, here physiological pH actually given and other kinds of microorganism actually given. Now, where we can use, what are the applications? First application is the transformation of steroids and sterol. Then if you have some non-steroidal compounds, then you can also use microbial biotransformation. For example, like dihydroxyacetone from glycerol you can get. You also can transform the prostaglandin, or you can also get the ascorbic acid also in antibiotic, pesticide, pollutants, or petroleum biotransformation can be done. Let me start with the steroid or sterols. So, steroids are actually natural compounds widely found in the bile salt, adrenaline cortex, sex hormone, alkaloids, or in some other kinds of antibiotic. So, progesterone, if you use this thing called rhizopus, this thing nigricans, you can transform this progesterone to hydroxyprogesterone. If you use some cyanobacterium complex with this thing called cortisol, you can get prednisolone. See, this is a pure example where we actually we have actually surpassed, or I can say we can get better result compared to our organic chemistry. We have used this thing called bacteria. Okay, we have used bacteria to get prednisolone. If I tell you more some of the bacterium complexes like Corynebacterium simplex, this thing actually dehydroxylate cortisone to produce prednisolone, whatever I have already told. It can bring about dehydrogenation of hydrocortisone or cortisol to produce prednisolone. Another thing I have to tell you that non-cardia restrictus biotransformation of this thing called cholesterol 9 hydroxy 3 1 into estrone. See, some of the names might be slightly typical, you should just remember for your just exam writing. Okay. So, you may just take screenshot, or you may just write down all this that is a typical name. I am going to tell you with the picture or diagram, I am not going to add, because if I add it will be huge long lecture, it will be more than 1.5 hour lecture. Then also this thing called androstenedione can be converted into testosterone by yeast. Again you can see some yeast is actually coming here. We have used some bacteria. Okay. So, everywhere actually using. Then Peterson and his associate also demonstrated that compounds like progesterone and this thing called resistant compound S may be biotransformed to testolectin through action of A flavors or P adametize. These are also some microorganism, again you can see. Okay, everywhere we are actually using microorganism, we are actually transforming something to something else. Okay. Here is a very big picture, again you might be so much that is curious, because you see here is progesterone, and every case we have used some microbes. We can use either bacteria, yeast or fungi, and ultimately we are getting some compound. Here we are getting one dehydro that is a testosterol lactone. Here we got the thing called testolectin. Here 6 beta hydroxy androstenedione 3,7 dione. Here we are getting del 1,4 andro this thing called stridone 3,1 dietone. Here again we are actually getting another substance called 4 androstenedione 3,17 dione. Similarly, you can see everywhere we are getting something new product. Okay, we are getting something new product. So, in such cases everywhere we are actually using the microbes. Now, what is the outcome of our entire learning? The outcome is that these microbes can bypass the general organic chemistry based work. In organic chemistry, what do we do? In organic chemistry, we actually synthesize these kinds of product. To synthesize this kinds of product, we have to see its this thing called scaffold. We have to see this scaffold, then we have to do some organic reaction, chain reaction from reaction 1, there will be some substance, then reaction 2, then reaction 3, then after that we will be getting our desired product. So, it will be time consuming. Sometimes it is not possible to synthesize a product by using the organic pathways, okay, inorganic that is by using organic chemistry. So, here you can use all those various microbes whatever I have listed out. Okay. Let me remove the ink. If you may take one screenshot, you can also post it in your study room. In future, you can also develop new technology, you can also develop new pathway, where if we synthesize a product through organic chemistry, it is taking so much long time, but in this case it will take so much less time. Here are few example, few of the product you can see and few of the manufacturer. Okay. This is not the fact that we are doing such kind of activity only in that is a research lab for literature. Actually, this process is so much popular and these things are actually widely used by various pharmaceutical or biotechnological company. These are the few are the manufacturing company like Upjohn, then Pfizer, then your ERP, Squab, Sons, then then your other organizations also have actually listed here. You can see everywhere they have used such microorganism to get this product. So, they have used this substrate and ultimately this is the reaction type. What kind of reaction type? They have used this kinds of substrate. Whatever I have mentioned you here, many of the products are you can see here. Okay. And this is the reaction type: 11 alpha hydroxylation, 11 beta hydroxylation, 16 alpha hydroxylation, 1 dehydrogenation, 1 dehydrogenation, side chain splitting. So, these are the type of reaction or this are the type of reaction what is happening with presence of such microbes, and ultimately you are getting this kinds of product from this. Okay. So, this is the something about the how you can get steroid or that is a steroid or sterile product. Now, another thing is actually non-steroidal product. For example, like dehydroxyacetone we can get from glycerol. So, dehydroxyacetone it is widely used in the lotion or various cream or cosmetics or various acetic acid bacteria actually we can use. For example, this thing called Gluconobacter melanogenous we can use. Then prostaglandin also, prostaglandin is also another kinds of non-steroidal compound. So, prostaglandins are produced from unsaturated fatty acids by microbial that is transformation of various kinds of fungi, okay, like Cryptococcus neoformans. Prostaglandin, why actually use this thing? This actually used as a contraceptive to reduce the pain during childbirth. For the treatment of also for congenital heart failure and also some digestive diseases, prostaglandin is actually widely used. If you use simple process organic chemistry, it will take so much time. Here you can use fungi. Then production of L-ascorbic acid, that is vitamin C. Vitamin C, all of you are aware why it is important. It is a good immune booster. You can use vitamin C to boost up your immunity. So, bioconversion of D-sorbitol to L-sorbase by Gluconobacter oxidans and several other chemical steps from L-sorbose to L-ascorbic acid. Ultimately, we are actually using some microbial organism for the to get our L-ascorbic acid, that is vitamin C. Now, in transformation of the antibiotics also, see many of the cases it is required that we have to transform our antibiotic. So, the biotransformation of D-sorbitol to L-sorbose by this kinds of bacteria, it is actually happening whatever I already covered. Now, here existing antibiotic leads to development of newer or modified or improved antibiotic, which contains many qualities like reduced toxicity, broad antimicrobial spectrum or enhanced oral absorption and less resistance or lesser allergic reaction. What are the examples? Like streptomycin. Okay. Streptomycin, Streptomyces parvulus produced new actinomycin with better pharmacological activity. Okay. So, if you use the macros, it will give you better results. Then also Lancashidin C is an antibiotic which was that is esterified to lancacidin C8 butyrate by microbe Bacillus megatherium. Okay. Then if I tell you more, lancacidin C8 butyrate has improved antimicrobial activity with the lesser side effect. In both these cases, we have better ultimately pharmacological activity, lesser side effects and improved that is a microbial activity. So, get better results we are actually using existing antibiotic, trying to that is make some change and get some better molecule which have better pharmacological effectivity. Also in pesticide also, pesticides are actually widely used to control the pest. Bioconversion leads to convert the pesticide which help to improve environmental condition. Now, why? Why, sir? It is very much important. You see, if a huge amounts of pesticide may accumulate in our soil, there are chances that our soil quality may get degrade. And if it is highly get deposited in our in our sea, if it is get deposited in high amount, then what will happen? Obviously, it will be harmful for our microbial environment present in our sea or ocean. So, that is why we have to also metabolize the undesired or if any high concentrated pesticides are present. So, in this case also microbes can help us. For example, conversion of pesticide or xenobiotics achieved in many way. Like here you can see this thing called Dalapon, it is a herbicide. So, here we are actually using Anthrobacter species and we are getting ultimately pyruvate. Okay. Then here you can see we have seen the metabolism. Another example let me take the this thing called cometabolism. So, we are actually using N-succina. Here is a molecule called 1,1,1 trichloro 2,2 bis 2 4 chlorophenyl ethene. This is actually DDT. Okay, sometimes we call it dichlorodiaphenyl ethane DDT. This DDT, all of you are aware, if high amount of DDT can that is accumulate in your soil, there are chances that soil quality might be get reduced. So, DDT can be converted into DDD 1,1 dichloro 2,2 bis 4 chlorophenyl ethane. Okay, we can get this thing by using N-succina. So, if I tell you some large scale conversation, large scale application where we actually using microbes. So, these are the few enzymes and we are actually doing that is a microbial biotransformation where such kind of that is a conversion actually happening, and you can see what is the large amount at the very large scale of product we are actually getting here. For example, amino this thing called acylase. So, by conversion of amino acid with the enzyme of this, you can see how vast amount of product we are actually getting. Another thing like penicillin acylase, you can use this enzyme. You can also use this thing called thermolysin, trypsin, various lipases. And ultimately, you can see how great amount of product we are actually getting. So, in large scale conversion, it is actually happening right now. Now, it is a growing field. At the beginning of my lecture, I have told you it is a very growing field. Now, why it is a very growing field? The answer is the ease with which the biological approach can be exploited with everyday organic, chemical or laboratory equipment. Then also the commercial availability of enzyme like Baker yeast or variety of microorganism is very easily commercially available. Then the ease with one can state away into complex chiral product for assessing their biological or industrial or medicinal utility. Generally, enzymes like biocatalysts are more efficient catalyst than chemical catalyst. Very low concentration actually required in this case for enzymatic reaction, where in other cases you need to the high that is amount. For chemical catalyst you need a very high amount. The availability of guides to procedure for a selection of a most suitable organism or desired conversion. Microbial biotransformation takes place under mild condition, often a room temperature or close to natural pH, thus minimizing the problem of isomerism, racemization, also equipmentization, epimerization and reagentment that often plague the traditional chemical method. See, here I have used some very tough word like isomerism. So, in our chemistry there is one subject called CADD, computer aided drug design. In CAD we are actually try to get new molecule. If we have one molecule, we try to make its multiple conformer and we try to see whether this conformer is fixing with our or matching with our target's active site or not. If it is matching very nicely, then that is fine, we are going to synthesize that product. But the problem is the temperature, or pH. You see, many of the conformer, many of the substances, in what conformer we have got it through our that is a CAD, computer drug design. That conformer might be not stable in normal room temperature or pH. That is why here we have to think alternatively. The answer is microbial biotransformation. Okay, also racemization, epimerization, rearrangement, and this all the things can be addressed by using microbial biotransformation. If I tell you more, enzyme systems are highly efficient, very selective in terms of type of reaction catalyzed and with respect to structure and stereochemistry of the substrate. Use of enzyme minimizing the desired, undesired side side reaction such as decomposition, isomerization, racemization and rearrangement can be a problem in chemical reaction. Then contrary to stereospecific enzymatic conversion, which are easy, the stereospecific chemical conversion of even a simple organic compound need a complex organometallic reagent, which is expensive and difficult to synthesize in simple laboratory and quite not possible for industry exploitation. Enzyme can be distinguished between enantiomer of a racemic substrate, therefore, only a enantiomer is actually attacked, leading to formation of one selective product. Can you see what are a great advantage of our microbial biotransformation? Only one enantiomer actually we are actually hitting. Then if I tell you more, the biotransformation exhibit reaction specificity, then also regospecificity, stereospecificity can be carried out under mild reaction condition. The substrate molecule is usually attacked at a specific site even if several groups of equipment or similar reactivity center are present. So, this is highly specific about these kinds of work. So, that is why it is getting so much popularity in recent times, and as a student of pharmaceutical biotechnology, I will also suggest that you may also try to explore. There are few other points: most biocatalyst, slow catalyst activity in both aqueous and organic solvent, hence suitable over conventional in chemical reaction. Enzymes, mediators of biotransformation are environmental friendly. They can degrade completely in environment, unlike chemical catalyst, which often require special procedure or processes to disposal. Okay. So, there are lots of advantages of such kinds of our activity. So, that is why we actually widely follow. So, today we have covered all the various aspects. We have seen that what is microbial biotransformation, how you can transform the steroid, how you can transform the non-steroid antibiotic, pesticide, pollutants and other that is petroleum also. So, in my next lecture I am going to tell you something very interesting: mutation. Mutation is very that is essential for today, because due to mutation we are observing a lots of variant of SARS-CoV-2. I am going to also touch the SARS-CoV-2 part. So, tomorrow we shall learn a new topic called mutation. I am going to tell you what is mutation, what are the types, diseases and what are the causes of mutation, and ultimately mutant by that is mutants arise by mutation. So, that is it from my side for today. I hope all of you have enjoyed this lecture, and I hope this lecture might have help you a lot to understanding our all the concepts. So, in next lecture we may learn something more. I will try to cover all the aspects of mutation, and that is it from my side for today. Thank you all of you for joining. We may meet in our next lecture. God bless you all. Thank you.